Welcome to this presentation on bacteria. The state objectives, they would like you to be able to describe how a disease is the result of a malfunctioning system, organ, and cell, and relate this to possible treatment interventions, for example, diabetes, cystic fibrosis, and lactose intolerance. But you also need to recognize that certain pathogens can seriously impair normal cell functions and the health of the organism. Emerging biotechnology shows promise in preventing and treating some of the diseases caused by bacteria. So some bacteria are very important, some bacteria are not very important. Some of the helpful bacteria, some of the things that we use them for, we use it for digestion. We do have bacteria in our colon and our gut, and it's very, very important to the survival, to the digestion of food, and therefore to the survival of the organism. Humans use bacteria in making food. We use it, for example, in yogurt. We use it to make cheese, so on and so forth. Bacteria is very helpful with decomposition. If there was no such thing as decomposition, there would be dead animals and plants all over the place, but because we have decomposition, bacteria that does decomposition, the animals and plants that have died previously are decomposed and their nutrients are recycled back into the environment. And lastly, the final example of helpful bacteria is plant growth. And bacteria in the ground, called nitrogen-fixing bacteria, will convert nitrogen in the air into a usable form of nitrogen for the plant. And the plant needs the nitrogen to function. Once it's in the plant, another animal or humans eat the plant, and that's how plants give their nitrogen to animals and humans. There, of course, are harmful bacteria, and I'm sure you are more familiar with these than helpful or good bacteria. And these are disease-causing, and some examples are food poisoning, tetanus, scarlet fever, whooping cough, strep throat, or tuberculosis, among many, many others. I encourage that if you don't know what any of these are, take a few minutes and look them up and find out about them. So something interesting that I just wanted to share with you is that 1 square inch of your skin is host to about 100,000 bacteria. They studied it, they've learned about it, and yes, that is the number. Within minutes of being born, you are covered in bacteria. They don't hurt you. They're actually helpful. A new, very recent finding has found that each person's belly button has over 2,000 different strains of bacteria. So I'm not talking about the number being 2,000. I'm talking 2,000 different strains of bacteria in each person's belly button. Basic bacterial structure, they have genetic material. Their genetic material is DNA. They're protected by a cell membrane. Sometimes, as you can see down here, they also have a cell wall. And they have ribosomes. They also have cytoplasm, and that is familiar to all living things, all living cells. Here you can see a picture. We've got the genetic material in the middle, specifically DNA. We've got a cell membrane with a cell wall, a cytoplasm, and of course ribosomes. And those are the 4 things common to all living things, minus the cell wall. Some bacteria have flagella, which help in movement. Some also have pili, and that also helps in movement. So I just want to quickly go over some of the ways that bacteria reproduce. The first way is called binary fission. This is asexual. And this is when a single cell divides into 2 identical new cells. And basically, very simply, it's mitosis. However, mutations do occur during reproduction of bacteria. So a lot of new forms emerge from binary fission, a lot of new forms of bacteria. Next is genetic recombination. There are 3 ways that prokaryotes or bacteria can form new genetic combinations. The first is conjugation, and this is when 2 bacteria exchange their genetic material. Next is transformation, and this occurs when bacteria take up DNA fragments from their environment. And we learned about transformation when we learned about Griffith. And lastly is transduction, and this occurs when genetic material, such as a plasmid, is transferred by a virus. And the plasmid often conveys antibiotic resistance. There's also something called endospore formation. And this is when some bacteria survive, they're able to survive harsh conditions by forming thick-walled structures called endospores. The endospore forms inside of the bacteria. They surround the DNA and a small bit of the cytoplasm. And then the endospore can survive boiling, radiation, acid, long periods of time, etc. They show no signs of life, and when conditions are favorable, they can basically be revived, come back to life hundreds of years later. Bacteria cause disease in one of 2 ways. The most common way that bacteria cause disease is by producing a poisonous chemical called toxins. The toxins may be released or stored inside the bacteria until the bacteria die. Foods contaminated with bacteria or with toxins can cause food poisoning. For example, botulism occurs when canned food is contaminated with endospores of bacteria. The bacteria multiply in the can, causing the toxins to build up. Botulinum, the name of the bacteria, botulinum toxin destroys the tips of nerve cells. Symptoms of botulism include double vision and paralysis. Botulism can be fatal if the muscles involved in breathing become paralyzed. Botulinum toxin is also now used in cosmetic procedures that paralyze nerves in the face and reduce wrinkling of the skin. The second way that bacteria cause disease is by producing enzymes that break down the host tissue into nutrients for the bacteria. A good example is tuberculosis. This is when a bacterium, specifically tuberculosis bacterium, uses the human tissue for nutrients. Some bacteria only cause mild illness, such as strep throat. Antibiotics are chemicals that inhibit the growth or kill microorganisms. Since the introduction of antibiotics, these drugs have reduced illnesses, suffering, and deaths from bacterial disease. Antibiotics only work on bacteria. They do not work on viruses. If you go to the doctor with a viral infection and they give you antibiotics, they will not work. They will not make you better. There is a concern, however, of antibiotic resistance. Over decades of antibiotic use and overuse, bacteria have developed resistance to these drugs. Remember, bacteria have the ability to mutate, and when they mutate, they become antibiotic resistant. And that means that they can basically tolerate the antibiotics. There are many, many consequences to this, because basically if bacteria continue becoming resistant to antibiotics, eventually we're going to come across a bacteria that is deadly, however, antibiotic resistance, and we will have no defense against it. So take a few minutes, answer these questions on loose leaf paper. I'll be collecting them in class tomorrow.